We'll start with a little bit of introduction. So let me share my screen and we'll dive into the presentation. Okay, can you guys just give me an okay sign that you can see my screen? I think it's okay, good. So welcome to the first webinar of the Hack4Rare, the rare disease hackathon that is gonna kick off the end of the month, June 25th, and will last until July 30th, 2021. It's a collaboration between the Children's Tumor Foundation, MIT Hacking Medicine, Sage Bionetworks, P10 Hamartoma Tumor Syndrome Foundation, Rasopathies Network, and Research to the People. So we have a lot of content that's going on, it's coming up, and we are really, really happy that you actually took the time to be here. As a welcome, and of course, thank you for joining us for the Hack4Rare. We already have 154 registrants for this event, which is amazing. So please keep spreading the words, call your friends. I think it's going to be awesome. It's going to be an amazing experience working on rare diseases and working with so many experts, mentors, and different expertise across the globe. So today we're here for the first track, which is Neurofibromatosis intro webinar. And you will learn about an overview and challenges of NF1, overview and challenges of NF2 and schwannomatosis. We'll learn about the NF data portal. So you can go nfdataportal.org and the datasets you can use. But we also talk about how to interact with NF mentors and patients. And we'll also review the initial pain points in track 1 if we have time. Otherwise, this will be, taken care of in the other webinars. So first, before we move and introduce our speakers, I just want to thank our early sponsors and partners. There's a lot of connections here to be made, and thank you to the amazing sponsors. This actually couldn't happen. So Wuxi Aptech, Helix, AstraZeneca, DNA Nexus, Barsum, the RACI Conference Series, Sage Bionetworks, the NF Open Science Initiative, the NF Data Portal, the FNR Registry, but also Rare Base, Desmoid Tumor Research Foundation, and the Childhood Cancer Data Lab. These are all our partners, and there are more coming. So this is going to be a crowded list, but this also kind of tells you what kind of connections and companies will be involved actively throughout the event. And let's go to today's speakers. We'll have Nancy Ratner, professor, the Department of Pediatrics of Cincinnati Children's Hospital, and she will talk about NF1. Marco Giovannini, professor in residence at Head and Neck Surgery, UCLA. We'll learn from him about NF2 and schwannomatosis. Then Jineta Banerjee, senior scientist at Sage Bionetworks, will explore, what is available in terms of data sets and NF data that you can use in this event. My friend and excellent colleague, Delberg, that is helping us navigate patient mentors and advocate with RAINS, but so is super involved with a lot of initiatives. And that's me, the Chief Scientific Officer of the Children's Tumor Foundation, today your host for this webinar. So quickly, what's going to happen on this track? We have 3 prizes, like the other tracks as well, for first, second, and third place. But also, what is important is to mention that the Children's Tumor Foundation has committed for this event for a pool of $30,000 in incubation prizes. So please check the Hack4Rare rule pages to understand how to apply and get to those prizes. This is the first of our series of webinars, and this week we're going to have another one. Just take note of this change of time we have for Thursday, June 10. 9 p.m. Eastern time. We'll discuss with research to the people about the collaborative track on desmoid tumor patient case. And then we'll pick up again next week, Tuesday and Thursday, with P10 amartoma tumor syndrome and the Rasopathies network. And then the week of June 21st, we'll have the hackathon overview with MIT Hacking Medicine. We'll start problem pitching. So we'll start the session of ideas and brainstorming about what can be done. And then Friday, June 25th, 12 to 2 p.m. is 2 hours. We'll kick off, we'll welcome, we'll have some team formation activities, but we also do problem pitching. So this is going to be a very interactive, everybody can participate and pitch their problem for a minute. Make sure you follow the conversation. So refer back to hack4rare.org website for the latest updates on the agenda, new content, features, resources, data announcements that are coming, new prizes also, and list of pitches if you want to see what the pitches are. But also join the Hack4Rare Slack workspace. I dropped a link on the chat. I will drop it right after I pass the baton to Nancy, and that will be for team formation, discussion of topics, but also access to mentors, general questions, support, and so on. So join the webinar discussion on Slack in track 1NF. That's the channel that we have dedicated. But also all webinars will be available in a few days on Hack4Rare. So even this content is going to be recorded and it's going to be available for you offline. So last thing, you want to learn more after you probably, we picked your interest after today. Well, the NF conference is just around the corner. So next week, June 14, 16, you'll have the World Conference at your fingertip. So we are very delighted and happy to offer complimentary code HACK21 Hack to waive registration fees if you want to join and learn more about the event. So that concludes my initial introduction. So it's my pleasure to introduce you to Nancy Ratner, and she will explain and tell us a little bit about NF1, not everything, because that's a very long story, I'm sure. Thank you, Nancy.
Indeed, indeed. So in, I hope, under 15 minutes, I'm going to share some thoughts about what unanswered questions are in neurofibromatosis type 1. Okay. So first of all, here's my email. You're welcome to contact me if I don't answer whatever kinds of questions you have by the end of today. We understand that we're studying a rare disorder and that we're clearly, to date, the genetic and genomic analyses have provided quite remarkable understanding of human diseases, including NF1. Of the rare genetic diseases, NF1 is relatively common. It affects about 1 in 2,500 individuals, is the newest data from a study in Finland. So as I said, I'm going to try to emphasize some of the unresolved questions that I think genomic data and high content imaging data may be on the cusp of allowing us to answer, and so relevant to the hackathon. Okay. So first of all, I don't know the level of sophistication of most of the people on this call, so I'm going to start from the basics, and that is neurofibromatosis type 1 is a genetic disease. It's caused, to a first approximation, by complete loss of function of the NF1 gene and therefore complete loss of function of the NF1 protein. And we can use genomic analysis, that is analysis of the DNA, transcriptomic analysis, that is analysis of the RNA derived from that normal or mutated DNA. And finally, proteomics, that is analysis of the proteins that are derived from the translation of the messenger RNAs inside the cell. So in NF1, analysis of the mutant DNA has shown that there are lots and lots of different mutations in the NF1 gene, but there are very rare correlations between patient genotype and patient phenotype. There are a few examples, but to a first approximation, these can be ignored in the big picture. So loss of NF1 protein affects many different types of cells. It can occur at various times, and therefore it can cause many different disease manifestations. Many of these manifestations involve cells with loss of function in both copies of the NF1 gene. So some of the features of NF1 that are used for diagnosis are hyperpigmented spots on the iris, the presence of hyperpigmented spots in the skin, freckling in the groin, bowing, for example, of the long bones or spinal scoliosis. And then tumors, for example, peripheral nerve tumors that we call neurofibromas. Now, when you ask patients or families or physicians, what are the most important problems that degrade the quality of life in individuals with NF1? The answers are very clear. Nerve tumors, which are neurofibromas and MPNST, which I'll discuss more in a bit. Brain tumors, which are largely low-grade gliomas called pilocytic astrocytoma, and these can lead to compromised vision. So while they are low grade, they can really, as I said, degrade quality of life. And finally, learning and attention problems, so cognitive deficits in NF1 that are of kind of understood, that are understood, yes. Okay, so one of the most intriguing and confusing features of NF1 disease is that the manifestations of NF1, that is the spectrum of phenotypes in an individual, can be very varied. So even in families in which all the individuals have the same inherited NF1 mutation, the same NF1 loss of function, some individuals can have many, many tumors or learning problems or so on, while others show only, for example, cafe au lait macules. It is believed that the variable manifestations may be due to the different expression of so-called modifier genes. Now, modifier genes are defined as genes that affect phenotype or expression of other genes. In other words, modifier genes can modify the outcome of having an NF1 mutation. And we believe that modifier genes are important because evidence from studies in which sets of monozygotic twins were compared to more distant relatives suggested that the twins were more like each other in their phenotypes and like more distant family members. And modifier genes have been implicated in numbers of dermal and plexiform neurofibromas and in whether or not an individual will derive a brain tumor. And so the first question I'm going to raise is whether we can actually identify modifier gene candidates in the genomic DNA in large data sets. And we need to answer questions like which modifiers exist for each distinct manifestation of NF1? How many modifiers are there? Do they overlap? You know, what about modifiers in glioma, dermal neurofibromas, attention, learning, plexiform neurofibroma? So with that, there's only a couple of instances in which modifiers have been studied. I'm going to start with the nerve tumors in NF1, that is dermal and plexiform neurofibromas. These are the benign tumors. Here's a picture of an individual with lots and lots of dermal neurofibromas. These are tumors on the skin. In imaging, you can see that they appear as these bright spots. Now, here's an example. And almost all, 95% of individuals with NF1 develop these dermal neurofibromas. Plexiform neurofibromas are now thought to develop in at least half of individuals with NF1. Here's one such tumor shown on a little boy. And here in an MRI image, you see this is a huge tumor. So these tumors can get very, very large, up to kilos in size, and really do significant damage when they compress, for example, the brain, the spinal cord, and other vital organs. These tumors have what is on imaging a little bright spot that is sort of a residual normal nerve surrounded by tumor. It is now known, okay, so in dermal and plexiform neurofibromas, NF1 mutation / loss is the only genetic change. And now numerous studies have looked for additional changes, and at least at the, with the sample numbers that have been evaluated in the kinds of ways they have been evaluated, NF1 is the only consistent genetic change. A little bit more aggressive is believed to be the atypical neurofibroma. Now, this tumor, in which the cells, this is a histology image, the tumors look in general a little more cellular. They have loss of NF1 and additional loss of CDKN2A, another tumor suppressor gene. But on imaging, they're basically impossible to tell from plexiform neurofibromas. So this is another area where imaging could possibly help to distinguish tumors. Now, these tumors themselves can transform into a very aggressive sarcoma called malignant peripheral nerve sheath tumors that are very frequently, most often lethal. And here again, the imaging, it would be really nice to be able to distinguish these two tumor types, but that this cannot yet be done accurately. And you can see on a histology level that you can see there are lots and lots and lots of cells here as compared to the lower grade tumors. So what are the questions to do with neurofibromas? I think one of the exciting things that's happened in the last few years is that the FDA has approved the use of a drug therapy, MEK inhibitors in NF1. But in some patients, the tumors don't shrink in response to MEK inhibitor, and we don't know why not. We think there should be other drugs that could act in concert with MEK inhibitor therapy for better outcomes, but we have not yet identified them. And finally, there are some people who feel that neurofibromas in different locations might differ from one another. And really, there's no definitive evidence. And we wonder if higher resolution studies from imaging could potentially, or genomics, could potentially identify those if they exist. So we ultimately, we want to know whether there are genotype or RNA features that are closely associated with the different tumor types, and wonder if we could use these features to identify new drug targets. Okay, coming back to modifiers, I just want to say that in NF1, my lab in particular has been quite interested in this. And there have been 3 studies that have looked at genomic features of NF1 patients and patient tumors. They're listed here. And when we collected all of them together, we identified changes, that is germline variants, that were present in increased numbers in these cohorts. And when one looks at the expression patterns of some of these genes, you can see that the expression of some of them is actually reduced compared to normal. And we focused on the ATM gene. It's published. I'm not going to belabor this, but I think at least this is some evidence that there are germline variants. These may modify NF1 phenotypes. So as I said, the malignancy that derives from neurofibroma is called a malignant peripheral nerve sheath tumor, and that these are lethal sarcomas, a major cause of death in adults with NF1. And they often, but not always, form in existing plexiform neurofibromas. And just to reiterate what I said a moment ago, many clinicians would really like to be able to identify aspects of images that would identify plexiform neurofibromas that will become atypical neurofibromas, or even more important, atypical neurofibromas that will become deadly MPNST. We don't know if there might be subtypes of MPNST. We know that there are huge numbers of gene mutations in MPNST. We know that there's gain and loss of certain tumor suppressors in MPNST, but they seem quite variable. And most important is, can we identify potential therapies for MPNST using the existing data sets? Now, I want to briefly mention brain tumors in NF1. I'm showing here at the end of the yellow arrow an image of one of these benign optic pathway gliomas. This is really a swelling of the nerve tract or optic nerve often, although these tumors can occur in other places in the brain. Very recently in April, a new data set was published which integrates clinical data, histology, and so on 70 individuals. And like neurofibromas, mainly they show only mutations in the NF1 gene, although some, which may be the more aggressive ones, have an additional gene change. So again, we're coming back to the same themes. Can we identify candidate therapies for NF1 using genomic data? Why do some, that is 15% of kids, develop these tumors, but not all of them, i.e. are there relevant modifier genes that we can identify? And can we accurately distinguish these tumors, wherever they are in the brain, from something else that occurs in imaging, which is these bright spots, which may be changes in glial cells? And that's, again, something I'm particularly interested in. Okay. And finally, in terms of NF1 manifestations to think about, there are these learning and attention problems. Some studies suggest that changes in that are detectable on brain imaging, MRI, diffuse and tensor imaging, or functional MRI correlate with these problems. And again, only half of individuals with NF1 have attention and learning problems, others don't. Why? Are there modifier genes? And one of the things we actually don't know, which is really amazing in this day and age, is whether unlike for the tumors, where complete loss of NF1 function is clearly necessary for tumor formation, in the case of these learning and attention problems, it still remains unclear whether you need complete loss of NF1 function or whether the partial loss of NF1 function is sufficient to generate phenotypes. And the very last point I want to make is that it is quite clear that a major limitation of studying rare diseases, including NF1, is that the number of studies of each disease are few, and the numbers of samples that are analyzed per study is also few, typically. However, I think that people in the field believe that there's a huge treasure trove of information that's maybe useful here. And that is, in adult studies, thousands of tumors have been analyzed. And we now know that in many of these adult cancers, there are NF1 mutations. So I queried a website called CBioPortal yesterday, and I found that in just these 5 studies that I queried, 142,000 tumors had been looked at, and a full 6% of them, or 824, had NF1 mutations, structural variants, amplifications, etc. And as you can see, in some kinds of tumors, I think this line is glioma, there are quite robust percent of tumors with NF1 mutations. And it is likely that these data sets could be leveraged to identify perhaps drug targets or features that correlate with loss of NF1. So that's what I said here. What genomic or transcriptomic features are most closely associated with NF1 tumors themselves, or with tumors with NF1 mutations in these sporadic cancers that could, in either case, help us to identify new drug targets to actually cure NF1. And finally, can we develop algorithms to use imaging to identify individuals with attention defects, learning problems, or predisposition to tumors that could help to better stratify individuals for trials and read out trial success. And I think I'll stop there and hope that you enjoyed that quick tour.
I definitely did, Nancy. Thank you.
But you already knew everything.
I know, yes, I know, but I think you actually put it in a very nice way, because we need those questions so that people on the call can actually, identify maybe ideas for, starting their project and looking at all of that massive amount of data that is out there. Okay, we are right on time. Thank you, Nancy. And we'll pick up any questions that are coming up. So folks, please type any question for Nancy. If you want her to address now, we can also, do it later. But I want now to introduce Marco Giovannini at UCLA. Will cover NF2 and schwannomatosis. Marco.
Yeah. Thank you, Salvo. Hello, everybody. Welcome. I will do a quick overview of the other side of neurofibromatosis, which is the neurofibromatosis type 2 and schwannomatosis. This is my email, so feel free to reach out and if you have any questions. We just heard about neurofibromatosis type 1 from Nancy, and I will focus on the type 2, what is called the type 2, and schwannomatosis, or type 3. So what is important to consider is that these are 3 different diseases caused by mutations in different genes. So the genes were identified for NF1 in 91, NF2 in 92, and schwannomatosis is way more recent, 2007 and 2014. So this is, another way of showing the molecular pathways leading to the different type of neurofibromatosis. So the common denominator is that the cell type, which is affected by the tumorigenic conversion and process, is the Schwann cell. And so this is common to all three types of neurofibromatosis. In the case of NF1, as we just heard, the NF1 gene is mutated in the germline of patients with a double inactivation in the tumors of those patients, leading to neurofibroma. And when additional mutations are combined with loss of NF1, we see malignant transformation of those otherwise benign tumor into MPNST. As far as schwannomatosis and NF2, the tumors that we see are slightly different from neurofibromas, and they're called schwannomas. So histologically, they are much more homogeneous, and they appear like a homogeneous population of tumoral Schwann cells. Very important is the vast majority of schwannomas in NF2 and schwannomatosis are benign. So these tumors grow locally, they do not invade, they don't give metastasis, and the only occasions where schwannomas progress into MPNSTs are in NF2 is when these tumors, okay, I cannot go back, here. When these tumors are irradiated, or in the case of schwannomatosis, the spontaneous transformation into MPNST has been sometimes described in a couple of families. So what are the major manifestations of neurofibromatosis type 2? Vestibular schwannoma, so a schwannoma of the 8th cranial nerve, the acoustic nerve, is seen bilateral in more than 95% of NF2 patients. So this is actually the hallmark of the disease. And genetic analysis shows that in those tumors, the NF2 gene is biallelically, both alleles of the NF2 genes are mutated. So these tumors do not express the NF2 protein, also called Merlin. About 50% of NF2 patients also develop multiple meningiomas. This is an example of a meningioma of a convexity in this patient. And this patient, unfortunately, also carried an ependymoma. This is another type of tumors that is associated with NF2 in about 5% of the patients. In all cases, tumor cells show the biallelic inactivation of the NF2 gene. So using mouse model, we have shown that indeed the biallelic loss of NF2 is necessary, is sufficient for tumor formation in mice as in humans. The NF2 gene is also mutated in sporadic tumor, as we just heard for NF1. And so we see it mutated in the same tumors type that we see in NF2 patients, but also in other tumors such as malignant mesothelioma, which is a tumor predisposed by asbestos exposure, and others, including breast cancers and melanoma, but they're very rare. So the clinical features of NF2, I just mentioned that the predominant manifestation is the presence of bilateral vestibular schwannoma, usually developed by 30 years of age. There are schwannomas of other cranial and spinal tumors and spinal nerves. As well as intracranial meningiomas ranging from 45 to 77%, depends on the case reports. Other manifestations of NF2 include cataracts. These are very important because they are an early diagnostic sign of NF2, being present even in children before tumor development is detected and obvious. Skin lesions, but less severe than in NF1. So what is important is that today there is no FDA-approved drug therapy for NF2, and surgical resection is the mainstay treatment for these tumors. And of course, surgical resection comes with the potential of neurologic deficits induced by, the surgical act, by removing those tumors. So especially in the case of vestibular schwannomas, deafness is a potential complication, facial paralysis, as well as lower cranial nerve deficits. Radiation therapy is generally not recommended for NF2 tumors, mainly because it doesn't work as well as for sporadic vestibular schwannoma, challenges in hearing preservation after irradiations, and most importantly, the risk of malignant transformation, as I already mentioned. So the medical treatments for NF2, again, no FDA-approved medical treatments. Chemotherapy is not effective. The reason is that these tumors have a very low proliferative index, so drugs attacking DNA and interfering with cell division are usually non-effective for these tumors. So the big question is, are molecular targeted therapies an option, a viable option for NF2? There are now 3 drugs, Avastin, which is an anti-VEGF antibody, everolimus, mTOR inhibitor, and lapatinib, an EGF inhibitor, that are used off-label. There are several studies showing partial efficacy of these drugs in controlling tumor growth and development, and particularly the interest of using Avastin for restoring failing hearing in NF2 patients. There is an ongoing trial with brigatinib. Brigatinib is a multi-kinase inhibitor, and this is an ongoing trial as part of the Intuit trial effort led by the Children's Tumor Foundation. So the question is, are gene therapy and immunotherapy potentially viable therapeutic venues for NF2? We don't know. So why it is so difficult to target the relevant cell signaling pathways underlying NF2 tumorigenesis. The NF2 gene encodes a protein called Merlin that function as a switch downstream of cell adhesion and RTK receptors at the cell surface. So the main issue is that Merlin activates several mitogenic pathways. So it remains unclear which one of these pathways are critical for tumor development. And so again, just a different way of saying the same thing is that NF2 Merlin controls multiple pathways that are all necessary for cell growth and are known to be associated with tumor development in other tumors and cancers. What happens is that loss of NF2 allows those pathways to go unchecked. And as a general consideration, treatments can be directed against specific proteins in these pathways in an effort to control tumor development. So schwannomatosis is the third form of neurofibromatosis. And it's characterized by the development of multiple schwannomas in the absence of bilateral vestibular schwannoma. So that's what differentiates NF2 and schwannomatosis. Bilateral vestibular schwannoma in NF2, no bilateral vestibular schwannomas in schwannomatosis. And the most common schwannoma locations, in contrast, are the extremities, spinal roots, chest, abdomen, pelvis, cranial nerves. Only 5% of meningioma, remember it was about 50% in NF2, of schwannomatosis patients develop meningioma. And what differentiates NF2 and schwannomatosis is that pain is often associated with tumor development or even in the absence of tumor development in schwannomatosis patients, and it's a common presenting feature of this disease. The genes for schwannomatosis that have been identified so far are SMARTB1 and LZTR1. So the mutations in SMARTB1 have been found in about 50% of familial schwannomatosis cases, and 20% of those cases, familial cases, carry LZTR1 mutations. What is very important, and I will stress this observation because I think it's the key to understand NF2 and the link between NF2 and schwannomatosis, is that mutation in SMARTB1 or LZTR1 and NF2 genes have been found in the majority of schwannoma in these schwannomatosis patients. So that means that schwannomatosis patients carry germline mutation of SMARTB1 or LZTR1, and this creates a mechanism of loss of heterozygosity, which removes the one by one, the two copies of the NF2 gene. So this is called the 4-hit 3-step mechanism of SMARTB1 NF2 gene inactivation or LZTR1 NF2 gene inactivation. And the reason for this complicated mechanism is that SMARTB1, LZTR1, and NF2 are closely located on chromosome 22. So loss of chromosome of this arm of chromosome 22 results in loss of the second copy of SMARTB1, one copy of NF2, and eventually an additional somatic mutation of NF2 completely removes the protein and leads to schwannoma development. So we have confirmed that this indeed what's happening by using a mouse model where we showed that mice with only SMARTB1 loss do not develop schwannoma, while mice carrying both SMARTB1 and NF2 loss develop schwannoma, as do mice with NF2 loss alone. So the conclusion is that NF2 loss is necessary and sufficient for schwannoma development, with important therapeutic implications. obviously. And this is quite surprising if we consider what is known about SMARB1, for example, which interacts with key proteins in pathways related to tumor proliferation and progression, such as the RB pathway, the Wnt signaling pathway, and LZTR1, which has been shown to be linked and activate, when lost, activate the RAS signaling, which is the one also altered in NF1. So this is my take on what are the crucial questions in the NF2 schwannomatosis field. Are the drug targets tumor specific? We now know from the studies that have been done with Avastin, everolimus, and lapatinib that there is an important heterogeneity, clinical heterogeneity of response to those drug interventions, suggesting inter and intra-tumor heterogeneity. So what are the therapeutic implications of tumor molecular heterogeneity, and how do we identify individualized treatment options, for example, omics analysis of tumor biopsies, and are single drugs or sufficient or drug combination necessary to combat these tumors, or alternate drug regimens? And how can we combat pain in schwannomatosis? And what are the molecular mechanisms of pain in schwannomatosis? So I stress the point that NF2 loss is sufficient for schwannoma development in schwannomatosis, but perhaps the mutation in SMARB1 and LZTR1, in addition to NF2, play a role in pain mechanisms in schwannomatosis. And finally, what are potential therapeutic venues for NF2 in schwannomatosis, gene therapy or immunotherapy? So I will stop here. I don't know if we are supposed to take questions.
Yeah, Marco, thank you so much. I think we have a couple of questions for Nancy from Sabina, and she was asking, where do you get the data from? I guess we can answer also with Jineta, presentation, but do you want to address that, Nancy?
Where do we get the data from?
Yeah.
Well,
I mean, is there any specific source, maybe that CBioPortal that you were showing?
CBioPortal is the place to go for the sporadic tumor information. There are several data sets deposited on Geo. Our data, for example, our transcriptome and genome data are on Geo. And then I think you're going to hear how Sage has collected a substantial amount of the data, ours and other people's, into this one site that you're going to hear about shortly.
Thank you. And for maybe for Marco, we have a few other things. So is there a treatment for NF2 approved in other countries?
No.
Okay. And then another form, I think, John, a few companies are exploring YAP-TEAD inhibitors. Are these being preclinically evaluated for NF2, or are they considered too toxic?
I was just looking at the agenda of the upcoming CTF conference, and there is an abstract. There will be a oral presentation presenting preclinical data in vitro and in vivo on the use of a TEAD inhibitor for NF2. So I don't know, but obviously someone is working on it, and we are very interested and excited to see the results.
Absolutely. So how does NF2 manifest in cataracts? This is a question from Chelsea.
In what? Sorry?
Cataracts.
Oh, interesting. There are a couple of papers. old papers now studying the mechanism of cataract formation in NF2 deficient mice. So these are mice where the NF2 gene was specifically mutated in the lens cells. And so basically, the idea is that Merlin has an important role in controlling cell polarity, and the orientation and the distribution of the cells in the lens is critical to give transparency to the lens as the bottom line. So if that precise parallel distribution of cells is not achieved because of Merlin loss, that could result in cataract. So this is a very, lay explanation, but I think that's the idea. And again, it seems to be a widespread manifestation of NF2. And as I said, depending on the studies, up to 90% of children with NF2 showed cataract or other eye abnormalities. And that was the actual diagnostic. So even in the absence of tumors, so before they develop tumors.
Nancy or Marco, feel free to chime in. Does the pain from Simone, does the pain have any particular characteristic that could help patients to describe, therefore raise a flag for diagnosis?
Well, in NF1, the answer is not really. I don't know how you would answer that, Marco, for NF2 or schwannomatosis.
Well, for schwannomatosis, what we know is that pain, so we are speaking of neuropathic pain, and pain can be associated with tumors. So subcutaneous tumors in masses in schwannomatosis patients can actually be painful, but it's not always the case. It can also be a diffuse neuropathic pain in a leg, in the arm, or somewhere in the trunk. So usually it's easy to make the association between pain and schwannomatosis when the tumor is painful. In the absence of an obvious painful tumor, you have to go through the diagnostic, the differential diagnostic checklist to differentiate from other peripheral neuropathies.
Okay.
But excellent question.
Thank you so much. And we'll move forward with Jineta. So there are a few questions about data, and I hope, Jineta, you'll be able to answer those. At least, giving some hints of what can be used during the hackathon. And then stick with us, because we also have Dale that will talk about, his experience last year and how to use patient representatives and patient mentors at the hackathon. We are a little bit over time, but we hope we'll just, wrap it up maybe 15 minutes together. So thank you so much, Jineta.
Thanks, Salvo. Yes, I'll definitely try to answer the data-related questions as much as we can. So I'll start off by showing what are the data sets that are available in the hackathon, and hopefully those will kind of point you to some of the data sets that can be useful for answering some questions that were posed today by Nancy and Marco. So if you have not registered already, please go ahead and register for the hackathon. And this is the main website for the hackathon, where you can find various information regarding the rules, as well as any other information about the team's data and the challenges that exist. If you are interested in finding out what data is available, please navigate to the data tab, or the data pull down list. And then here, what you'll see is a whole list of resources that host data that can be useful for answering various questions that have come up today and may come up during your participation in the hackathon. I want to specifically mention the NF data portal, because this data portal has data that is specifically geared towards neurofibromatosis. So if you were to look for more data regarding questions in neurofibromatosis, I would recommend looking here. So once you open the data portal, you will land up in the page, the home page of the NF data portal, where you'll find there are various things like studies, files, publications, and tools that can be of use for this, for your work. We would recommend exploring the studies because there you'll find many studies that have been shared with the research, with the larger community by researchers who have generated their own data and have been very, very generous in sharing the data with the research community as well as the larger public. So let's assume that you have a question which you want to answer by exploring some, let's say, gene expression data, for example, like RNA-seq data. So what I would recommend here is to kind of navigate to the data type for these studies and filter the studies that have gene expression data. And once you select gene expression in the data type on the left-hand side tab, you will see the list of studies becomes filtered onto studies that have gene expression data in them. So this is a list of all the studies that have gene expression data. And let me highlight one study here, which is the cutaneous neurofibroma data resource. And let's say that I want to explore what kind of gene expression data is available in this study. So I can go to the study page. There will be some information about the study itself, more details down below. I would point your attention to the access requirements. So many of the studies that have data that have been shared with the public, they sometimes contain sensitive data, and hence they might require requesting for access to that data. So please check what are the access requirements for the studies that you're interested in. And as soon as you see that you have to request an access for this data set, we would recommend doing that as early as possible because the access requirements sometimes take a little time. So go ahead and request access, and this will navigate you to its own original study page. So if you were to go to that study page, you would get more information about how to access the data or how to request the access to this data. So for example, in this case, you have to actually enter some information to describe why you want to access this data and why you need the data for a project that you're interested in. In this case, I had already requested access to the data. So my access was granted, and that is being shown here. So as soon as your access is granted to any of these data sets, you would get a notification saying that your access has been granted. So coming back to, coming back to the NF data portal again, once you have requested access, you can also go to the study data tab and explore more in granularity what kind of files are available for download. So let's say that I am interested in gene expression data, so I would specifically filter all the files by RNA-seq, and you can use either the left-hand side table to select these different kinds of data types, or you can also select them by clicking on the pie charts right here. So once you select the RNA-seq data, as I have done here, you can also select the various different file formats that are available for the data types. So let's say that I want to select only the raw data files like FASTQ files. I would click on this, and this will basically filter my data set or my the available files by the specific data type as well as the specific file format. So once I have really narrowed down what are the files I'm interested in, I can go ahead and download these data by clicking on this arrow, the download arrow right here. And it gives me 3 different ways to download the data. And for any number of files that are above 100, we would recommend using the programmatic option, which gives you specific instructions of how to do that. And once you have access granted to this data, you would be able to use these instructions to download the data on your either instance or your local machine. So now that now that you know, first of all, where to go to find various data sets, and also how to download some of the data files that you might be interested in, let me go back to the Hack4Rare website and go to the challenge track. And in the NF or the neurofibromatosis challenge track, we have also, if you scroll down all the way to the bottom to the resources, we have also started to provide a quick link to some of the highlighted data sets. And here you can see the various types of data sets, some examples of various types of data sets that can be useful for answering questions that may come up in this challenge track. And we also have, for example, for genomics data, we have some additional code snippets and additional information to help you kind of get started on your journey towards exploring these data sets. The quick link is still under construction. So you might not have access to it right away, but you will have access to it very, very soon. So, and I also wanted to say that there are new data sets that are also gonna come in soon and add to the data sets that are already available on the NF data portal. So I would encourage you to come and check on this, on the challenge track page, resources tab, to find more and more information about data sets that are available. So with that, I'm gonna
Thank you, Jineta. Yes. So question is, we'll post more and more stuff. So keep coming back to the pages so that you will have access to the latest, data sets and resources available. And let me jump now to Dale. Dale, thank you so much for joining us. You've been one of the highlights of last year's. You've been a patient representative and mentor. Tell us about your experience and how attendees can actually use you as a resource for their project. Because most of them, they say, "Oh, I don't have a medical background," but there are, and I don't know, maybe to use healthcare, scientific data, but there are so many other things to work on, right? And so many other problems that they're not just genomics.
Yeah, that was quite a deep dive, the first 3 presentations. So thank you for that. I'm in the consumer retail business. I try to find the latest and greatest product, to be able to sell to Target and to Best Buy, and seen innovation over the last 25 plus years in the retail business. And it's been sort of exciting as a schwannomatosis patient. join the CTF, in many ways, and the hackathon to kick around ideas and see how what's being worked on in your labs with your computer coding to find out what's going to be the next greatest hit in CTF. Whether it was in my world an MP3 player, a DVD player, a digital camera. We're here to just whether it's a minute or two, do not fret to engage us, because we're that early adopter. We're going to be the first one that's going to take that new car out for a test drive and or wear that wireless headset for the first time, or buy that first iPod. And really, I think that becomes exciting then from the participants here. You can sort of say, hey, I helped initiate that idea 5 years ago, 10 years ago, 20 years ago, and look how it revolutionized not maybe only NF, but other diseases, rare diseases. So, but it's fun. I look forward to it. It was like riding a bike the first time last year with the NF Hackathon. I learned a little bit. I'm hoping I got the training wheels off, and we'll be able to, as others, be able to help facilitate your thoughts and go for the gold. Let's go for the win.
Yeah, and sometimes understanding and framing the pain point where you want to focus. It's very useful to have a check-in with, of course, an expert, but also with a patient, because ultimately this research needs to provide some benefit at the end of the line, right? So the final user of these advancements in research are patients, and we need to have patients in mind and always connect the lines, even if it's basic research and you don't see how this can actually improve their life. But that's where your perspective is very, very important.
Yep. And others. And I've learned so much from other NF patients too. So we're all unique. And it definitely in the NF world, we got a slightly different story, and I think getting those ideas together can help this group move forward.
So folks, feel free to ping Dale. Is schwannomatosis patients, but was awesome. A lot of other talents and perspective on NF that can definitely give you some ideas, and we'll also list on the mentors page of the website other patients and other experts that can give you their perspective and their help. So thank you for being with us today. I think I also posted again the complimentary code to join the conference if you guys are. Okay, Sabina was saying, how do we get in touch with Dale? Dale is gonna be on the mentor channel on Slack. We'll make sure. So there are Slack channels that are made for interaction with mentors for each one of the tracks, but also, you can post any type of, question to mentors in the Slack channels, even in the all participants. We have people that monitor the chat and will address and make sure that you need one-on-one time, or someone actually answered that question, and Dale knows that the question is there. Super. Okay, thank you so much, and I hope I'll see some of you on Thursday and the next webinars. But please mark your calendar, June 21st and June 25th for the start of the Hackathon week. Thank you and have a great day. Thanks, guys.
Thank you, everyone.